Hello, listeners. Yamina here. Welcome to this very special resilience episode of the Dr. GPCR podcast. While we work on bringing you weekly new episodes of the podcast starting in September, here is a previously never released conversation I had with a previous podcast guest. Before jumping into this episode, I want to take a moment to talk to you about the upcoming Dr. GPCR Summit held between September 14 and 18, 2020. This summit is unlike any other meeting you've attended, and we are very excited about it. Our goal is to allow everyone in the GPCR community to get access to talks through the entire length of the event, independently of their time zone. This means that presenters will have the option of providing a pre-recorded talk or giving a live presentation. Think of it as a Netflix type of GPCR Summit. For more details, visit drgpcr.com/summit-2020. If you have any questions, please join our Dr. GPCR Club on LinkedIn. We also have a very special announcement to make. We are delighted to announce that we are joining forces with the Great Lakes GPCR Retreat to bring you closer to GPCR research during a live trainee event held on September 18th, 2020. And now, let's dive into our episode. Hi, Paul. Welcome back. I'm so happy to talk to you today.
Well, it's great to talk to you again, Yamina. So, as I was saying before you put the microphone on, my lab is sort of ground to a halt as far as actual experiments being conducted, other than tracking some animals that we're really interested in and that were sacrificed during this time and that have a kind of a very interesting phenotype. But otherwise, we are not able to really do any work, although, as I mentioned to you, I just learned I got a grant that's going to kick in July 1, so that will be very helpful. But what I actually have been working harder than ever, in fact, because, as I was about to say before, Krishna Sriraman, my lab, who's a postdoc I think you met when you visited once.
Yes, I have.
He called me up one day, and I was out. I know exactly where I was. I was going for a walk with my wife and dog, and he said, "Well, I was thinking about something related to COVID. I want to just bounce this off you." And we started talking, and one thing led to another, and before we knew it, we sort of decided we should write up something, an idea first, an analysis of what was then at the beginning a big deal about the use of angiotensin receptor blockers and ACE inhibitors, angiotensin converting enzyme inhibitors, as being dangerous. And I literally had gone to a talk that week from a leading guy who said he was going to stop his drugs, and another person I heard was going to stop their drugs. And we decided to look at the data really carefully in the literature on that, and decide. And when we did that, we realized that this was a bogus action, that the data did not support that these drugs were contributing to the worsening in patients who had hypertension or older patients and so forth. So we wrote that up, and it was published last month in the Clinical Pharmacology and Therapeutics. But then we wrote a very long paper proposing a hypothesis, which we've now realized multiple other people had at about the same time, that a major contributor to the pathobiology of the COVID-19 infection really is imbalance between angiotensin working through the angiotensin receptor 1 and the ACE2 that is generating angiotensin 1 to 7 and perhaps other entities, other peptides. And we wrote this into a very pretty big paper called Hypothesis of the Imbalance in Signaling as a key contributor, a central contributor to the biology. So, and I decided to send that to the British Journal of Pharmacology because they had just announced they were having a COVID call for papers, and it was accepted. From the day we sent it in, decision, revision, acceptance was 9 days, and I thought, and I was really pleased with that. And the paper's been online now for about a month.
Wow.
And since then, we've written a letter to the editor that appeared today in the American Journal of Physiology as a follow-up to this, and proposing that there should be benefit from this. And we hooked up with a guy here who was already doing a trial, I learned. And we're writing a perspective for the PNAS that we're supposed to send any day now as a follow-up to that too. But more than that, I've gotten calls from the popular press, and we've had a thing in Wired and something called The Conversation in CNN News. But the ultimate coup was about 2 weeks ago. Somebody had seen the figure in our paper in British Journal and from the Economist. And this writer called me and said, we'd like to use your figure, or wrote to me, we'd like to use your figure. Would that be okay? I said, of course. And then we revised the figure for them to make it a little simpler, a little more focused. We sent it to them, but we had done like, a classic, grade school picture, sort of, with arrows and boxes and things. And they turned it into a human body and put in organs and all this stuff. And it was a really sexy picture, really nice. And they sent it to me like 2 weeks ago. And I went over it. And then I actually edited the article for them because they made some mistakes, I thought, and errors. And it appeared on June the 6th with a nice little, attribution to us in the figure. So if you would have told me 3 months ago that I was going to end up having a figure in The Economist, forget about one of the science journals, in The Economist. And needless to say, everyone in my family is very impressed by this. So we've been very busy around this ACE. And then on top of that, I've just been asked to write the next chapter in the Goodman Gilman textbook on angiotensin signaling and angiotensin drugs. And I go, okay. So, it's kind of weird how things happen like that. I guess I've been able to, quote unquote, take advantage of this time to move in a little different direction. But as I think I wrote to you in an email, it's still about GPCRs.
It's always about GPCRs, even in The Economist, apparently.
Yes, it is. And so, we snuck that in a little bit in the article. I think we may have mentioned it. I can't recall for sure in The Economist. So the article appeared in last week's Economist.
Fantastic. I'm gonna take a look at that, and I'm definitely gonna link it to this episode's webpage. That's fascinating.
Honestly, this is so. As I said, I've been working really hard. We have another paper that has just essentially been accepted, but we have to just revise it. Again, I'm just doing British Journal because they're really fast and they're really interested in this, and related to an idea that we had about how it might be appropriate to think about repurposing a PAR1 inhibitor and think about PAR4 inhibitors for COVID based on effects on endothelial cells especially, but also on fibroblasts. And in terms of, again, what the really point I want to make about this, which hasn't really sunk into the broad society, everyone's talking about making vaccines. Yes, people are talking about antivirals, but there hasn't been that much effort emphasized about both treatment directly of the pathobiology, what doctors have to do to treat patients. A couple of drugs have been out around that, and a lot of people are talking about the cytokine storm and all the cytokines, and there are a lot of anti-cytokine things being tried. But coming upstream and thinking about ways to deal with the fact that ACE2 is being affected, but being bound, and one's getting stimulation by angiotensin and probably other peptides, and perhaps thrombosis is occurring and endothelitis is going on. There's a lot of really interesting opportunities. And so just as a follow-up on that, I don't want to say too much because this could be going public because we haven't written it yet, but we have an even broader idea around a therapeutic approaches that could be tried. But we're really intrigued by the possibility of repurposing drugs that really get at the pathobiology and that are involved in, let's say, GPCR activation and signaling. There's a, as you may know, and probably know because it's all been in the news the last 2 days, Eureka, dexamethasone, it works as an anti-inflammatory. Well, if you start looking at what dexamethasone does, that's probably anti-inflammatory. There's some very interesting other ways of going about doing what it may be doing. And we're going to write something up about that. It just sort of hit me on Monday when I saw that there's a GPCR targeted drug that's going into trial for COVID. And I looked at it even before I knew about dexamethasone. I was like, oh my gosh, I see exactly where this could be expanded in a much more scientific and broader way. And so I'm really excited about this. So I'm doing what I'm going to call armchair science. You know, we're not in the lab. This is really dry lab science, right? It's complete dry lab, but it's about when you can't do the work, you just think and read, and you know, and sometimes, as someone once told me when I was clinically active, don't just do something, stand there, you know, and it's that's sort of what we've been doing, and I think we've made some contributions that are pretty interesting. And I mean, I really want to do something to help people. That's what this is all about, really.
And sometimes it's important to stop and think about the problem itself instead of having your head in the wet lab all the time. What do you think about, one advantage of these drugs that already exist and repurposing them is that they must have went through testing, they're super safe. Do you see a cocktail of drugs being used to help alleviate the symptoms of COVID?
Yeah, I think it may be that you're going to need to use more than one, because I think different people are having different manifestations. And I don't know that, and I think there's multiple contributors to what we're calling the pathobiology of the disease. And I think you're going to have multiple targets that could impact on that pathobiology in useful ways. I mean, in the article, we've written about the ACE2, the imbalance in the angiotensin signaling. You know, you could say, well, when imbalance implies that there's two things working in opposite directions. And so you can either say, well, I've got to stimulate the thing that isn't working as well, and I got to block the thing that's working too much. So, you know, one of my philosophical points in science is that almost most biological systems are all about gas pedals and brakes. And, you know, we have proteases and we have kinases and phosphatases. We have cyclases that make cyclic AMP, and we have phosphodiesterase that degrades it. I mean, it's all over the place in biology, where we don't drive our cars with just our gas pedal, hopefully, and we certainly don't drive with our brakes. So if you think about it, nature has built in these systems consistently, and we by taking advantage of it, by either activating something that's good and blocking something that's bad. And in the British Journal paper, I wrote that section actually, described several several ways one might go about that. But now that I've been thinking about it more, I think there's even more ways you can do that in with respect to COVID. And I think you may need more than one, and you may need additivity. For example, it might be you're going to put dexamethasone with, let's say, a GPCR. Oh, gee, isn't that what they already do in asthma? Hello, that's what the therapy of these drugs that are on the market in asthma for, right? Long-acting beta blockers and glucocorticoids. Ding, ding, ding. I mean, that may be really useful. Keep in mind, interestingly, that the type 2 pneumocyte, which is the cell that gets hit in the lung that's got the ACE2, if I'm not mistaken, I was told 40 years ago by somebody who was going around assaying all kinds of cells, the lung and probably in the airway in the type 2 pneumocyte has the highest concentration of beta-2 receptors in the body. And so, and cyclic AMP, and that's why they give that to women with infantile respiratory distress syndrome, neonatal respiratory distress syndrome. That's why women are given steroids. That's why women are given beta agonists to help create surfactant. And surfactant is one of the big problems, the lack of that in the acute respiratory distress syndrome, which is what the patients have. So, I mean, you know, and it's pretty logical. This is not rocket science to me. It seems pretty straightforward. But people haven't been connecting some of the dots, I think, as pharmacologists, let's say, and as physiologists, actually. But anyway, I've been ranting on about this, but you can see I'm really enthusiastic about this thing. I think there's more to be done, because this disease, I think, is not going to go away, and it's going to be with us for a while. And I think there are people who are, the death rate's going to go up in the United States and in other countries. The deaths are, and especially in people of my age and older. And when you look at the average age of people who are dying is in their 60s, in their 70s, or even 80s. And so it's not clear why that is completely. There's probably many reasons, but I think we're going to need treatments for people, not just a vaccine and not just antivirals per se. But that's one. And I think there's a way where people who do the kind of stuff that we've done, that you do, that have been interested in let's say in the GPCR world and the signaling world, can really make a contribution this way. That's why I feel like I'm very enthusiastic about doing this.
So even
I may have said more than this.
Armchair science still is exciting, and it gives you time to think about these things. And I'm glad that you were able to publish and write all these papers in such a short period of time. When and if and when you're going back to the lab, are you planning on trying to understand these different signaling pathways in the
Yeah, actually, as I mentioned briefly, we I mean, the way these things happen is a little bit serendipity, but there's a guy at UCSD who's sort of been the major leader on the whole COVID-19 area. He was the head of infectious disease. He's he's a really terrific guy who's been involved in HIV and other viral things. So he's very knowledgeable. And I initially ran our idea by him when we first were writing up about the potential non-danger of ACE inhibitors. And he thought that was really very interesting. And I sent him a copy of the paper before it came out in British Journal. And he thought that was pretty cool. And then I said, well, I'm so excited about this. I think we should try to get a clinical trial going to think about giving these drugs. And he said, oh, do you know so-and-so on our faculty he's already doing a clinical trial with ACE inhibitors. He's just putting it into the FDA. And I didn't know that guy. I didn't know that guy personally, but we got in touch and we talked and did a Zoom or two. And before we knew it, I said, he said, maybe you guys can help me because I need help in assaying some of the samples that are going to come from the patients. And we said, sure, we could do those kind of assays, and told them about some of the assays we could run. So we may, when we go back in the lab, actually be assisting with that clinical trial that's being tried. Both here, he's got 15, if I'm not mistaken, he's going to have 600 patients and 15 sites, and it's a pretty big deal. But the center for the trials here, and he's hoping to involve a number of patients here. So he's not going to test all the patients' samples from other places, but the ones from here, hopefully we'll involve them. We haven't talked about that, but he's writing with us with his PNAS paper that I mentioned. And so we sent it to the draft of him on Friday, waiting to hear back from him. So I think, we may do some of that, but we'll probably also work on we got a grant not to do that. And we haven't put a grant in on COVID. I mean, we probably should have in retrospect, but I can't, we're just writing. We haven't thought about some more of that, but I think there's some really, I think part of the problem I would just say about COVID-19 I'm discovering is that none of the animal models that are accessible to us are all that good. Plus you're really supposed to work in a biosafety level 3 facility, and not everybody has those accessible. So it's not so easy. And I've actually been in touch with a couple of CROs about trying to get some stuff going on a related idea we've had for this, and they're completely backed up. And so many people are going to them because of that limitation. So it's going to be a little hard, I think, for academics to, they can do certain kinds of experiments, but the ones that look at what we're looking at, which is impact on the pathology and the disease, the mouse models are not ideal for that. And there's a model in the ferret, not completely ideal. There's one in hamsters, but I think that's going to be a bit of a problem. Again, not my area of expertise, obviously, but I'm learning fast, hopefully.
Hopefully the backing up in the CRO world where all the samples are going to get processed, and at some point you're going to get access to these. What kind of assays are you planning to do on the samples that you will get from this clinical trial?
Well, they're really going to be, things that relate to, the ones that have been assessed in patients as evidence of severity of disease. The hope is that that includes various cytokines, various markers of tissue damage, and possibly circulating ACE2 levels. All ones that seem directly linked to the disease pathogenesis and progression. The hope is that if you give these drugs, especially at an early stage, you can prevent progression. So you want to be able to have readouts that are biomarkers for the progression of the disease, and they include some of the things I mentioned, as D-dimer, as of the evidence of thrombosis and intravascular coagulation, perhaps, and some of the cytokines in particular. I mean, those are what seem to be factors in progression, as well as secondary infections that patients get.
And just to make sure I don't say something that's not quite right, we don't yet have an idea on these clinical markers that would predict disease progression if somebody would just get a quote unquote regular cold type of symptoms versus somebody who will end up on a ventilator.
My impression is that is correct, that people have found certain associations, but nothing with the absolute predictability based on what we know so far. And that's part of why people are collecting panels of these things to begin to say which ones might be best to do this. I mean, there's been a couple of studies I saw where people from China looked at a whole battery of things. Oh, these 4 things together seem to be a marker for severity and progression, but I don't know that that's been completely accepted yet. It hasn't been. I'm not sure it's been replicated. I'm not even sure it's actually published yet. And I think I'm not even sure. It may have been a preprint, but they did come up with some combination of markers that they thought might be predictive. But I don't know that that's been really well documented, confirmed, replicated, et cetera. So I still think we're at a discovery phase. Everything I've been reading, so people say this virus is one we don't fully understand yet, and I think that's why there's been such an effort to say, well, let's make a vaccine, let's stop it, But the question is, you got to do that in a time when lots of people are sick and people are dying. And so how much, another question, by the way, I should tell you that we came up against, and I think this is really important and exciting in a way, is some of the drugs that we are thinking about, much like what went on with hydroxychloroquine, which of course is now a bust, was how do you decide about taking an off-label use of a drug that, as you said earlier, has already been tested, can be repurposed potentially, because you have some idea of the safety in normal people or in people with certain diseases, but you don't know about exactly here. And the bottom line is, we're going to have to do randomized controlled trials of all these drugs to know for sure. However, I mean, how quickly can we get those data? Do we say, you know what, we have a really strong belief that some of these drugs, certain drugs might be beneficial. To our amazement, no one has ever put together, as we could find, criteria to make a decision about off-label use of drugs that takes into account the scientific questions and the ethical and the legal questions. So we decided to write a paper on that too. And I've got an ethicist to work with us on this. I've written to somebody at Stanford, a colleague, to see if he'll join, because he has contacts with people in the Stanford Law School about this, to come up with a kind of a consensus about how do you make a decision in the midst of a pandemic about possibly giving people a drug that has been tried and that has safety data, but hasn't been tried in this condition. And you know that they're going to have to be, the clinical trials are going to have to be done. But what criteria do you use to decide that you should go ahead and try it? It's as Trump advised, against all the medical information, by the way, of course, which turned out to be a bust. And we may contrast hydroxychloroquine with, let's say, ACE inhibitors and ARBs, where we think you can make a stronger case. Anyway, it's in the process of being written now, actually.
Wow. Yeah, and it's a big concern, and as you had mentioned, the safety in this particular case. It may be safe in the disease that that it's designed for, but who knows how patients who have been infected with the virus will react to it. There has been a couple of articles out there talking about looking for genetic determinants that make people more sensitive to the infection and to the complications that come with it. What are your thoughts about that?
Yeah, that's a great question. And we consider that in our paper in British Journal. Not that I'm pushing people read it, but I immediately thought of that too. Why, is there something different? And actually, one of the early findings was, that African Americans and some other groups were potentially ethnic groups, maybe at greater risk in terms of our vulnerability and severity and deaths. And so I immediately spent quite a bit of time looking up the data related to ACE2 on that and ACE1. And there is for ACE1 data suggesting that there is an alternative form of ACE that that does produce more angiotensin and therefore might be associated. But when I followed that up and looked at some pretty good population data that was assembled here in the United States about 15 years ago, the claim was it was statistically different. There was a slightly higher incidence of the more active isoform in African Americans than in Caucasians, but I think those differences those are mostly self-identified, of course, and that's also a little confusing sometimes. But as I recall, the difference was 32% versus 28%. And, looked at 5,000 people or whatever it was, several thousand people, I can't remember the number, that was statistically significant. But, I don't know, 28 to 32 doesn't strike me as a huge biologically significant difference.
Not a clear cut.
Huh?
Not a clear cut.
Not a clear cut. But the original data on it was very clear cut, but that was only after they did many more people when that I think it became more questionable. However, having said that, the possibility that there are other genetic factors, as you bring up, is a great point. I think one has to consider, is it a lot of the focus has been on the associated diseases that people have and the quality of their food and their lifestyle and all that. But I wonder if it'll turn out that there are some genetic determinants of severity that haven't yet been uncovered. And it will be interesting to see if that may exist in receptors or in peptide production or degradation or that kind of thing. That's a really important open question, I think.
And we need to learn more, especially when you look at all these young age children who end up catching the virus and ending up in hospitals and with this inflammation syndrome and Kawasaki-like disease.
You're right. What's different about them? And I worked with one of the women. We actually proposed a study some years ago. Who's like an international expert on Kawasaki's disease here at UCSD, and we proposed that there may be some alterations in certain of the GPCRs that might be predisposing to that. And we looked in the beta receptor for some reason. I don't even remember why anymore, but it didn't seem to hold up. So we never pursued that. But who knows? Maybe that's something like that is going on. And you're absolutely right in the kids. Why do certain kids get it and others don't? I mean, what are these factors? It's highly likely to me there's something genetic, I would think, going on, but we don't know yet for sure.
I think the world was taken by the whole shocking devastation that the COVID-19 disease, virus brought in, and we're just trying to catch up on right. Everything at the same time. We were talking about developing a vaccine versus repurposing drugs. When we look at the vaccine and development of a vaccine, everything is speeded up at this moment. What do you think about speeding up, this whole process of getting a vaccine from, for example, 5 years to 1 year? Do you think,
Well, I think they have to try. I mean, I think I just read something today that many of the companies who are working on this are already gearing up for their production side, looking to see what was the outcome of people who got that. So to the extent the virus is still, you need to have a virus still active in the community to be able to assess the efficacy of a vaccine. And how long are you going to take to do that? And when you have to take a group of people and say some are going to get a placebo, some are going to get antibody, or excuse me, the vaccine, and then wait and see what happens over some period of time. So if you just think about the logistics of that, it's going to take a little bit of time to do that.
It will. It will. And I was talking to someone earlier today, and the person said, well, I know they're working very hard to get a vaccine out so that people can get it. But this person told me, I'm not sure I would want to get the vaccine as it comes out fresh, because everything is speeded up, and how secure is it, and how efficacious is it?
Yeah, it's not clear. It's not clear. I mean, basically, they're going to test it in vitro, for blocking the virus or where it may work and preventing infections. Ultimately, in animal models, one would imagine, and ideally in non-human primates, which are a pretty good model, but that's an expensive model of any disease. And then once that looks valid, because the non-human primate that I've seen, there's a paper, I think, in Nature, I think it's out now, it was in press a couple of weeks ago, showing a beautiful picture in some non-human primates. I can't remember which group of monkeys, it's on the tip of my tongue, but I've forgotten which one for sure, that has a syndrome very similar to our lung involvement and so forth. And so that's probably how you're going to have to at least test it for efficacy early on. And then you're going to hopefully move in with that works, and you're going to have to go into clinical trials. So, I mean, I don't know how soon it's going to, it might be possible they'll have something by early next year. I heard that at least one trial is beginning this month, and another one may be beginning next month, and that's fantastic, But
it is, and there's 135 variations of the vaccine that are competing.
That's more than I had heard. It was only 120. Someone told me today it was 70. Now I said 120. You said 135. I don't know. It's something like that.
Yes, hopefully one of them will be safe, will be effective, and we can start immunizing people against COVID.
Well, I think many people believe that they're not going to really. Everything's not going to go back to being like it was, even though we probably never will go back like it was completely. But that certain activities will probably not really be entered into by everyone until there's a vaccine. I think whether it be attending to sports events or theaters or other activities, I think some people are going to be really gun shy to do that until there's really a sense of safety in the broader population.
Definitely. Even kids going back to school in September is still a question mark
Right.
on how many kids can be in a class, and all the safety protocols that have to be put together around the kids to make sure that they don't get it, they don't spread it, and the teachers also are safe. It's a new world. Since we last spoke in February, it's a whole different world.
Yeah, I forgot when we had spoken, but yeah, I guess February, so much has changed. It's happening at warp speed. That's what they're calling the vaccine program. This is warp speed in our own lives, I think. Along with all of the other things that have been going on around the impact on the economy, and now the Black Lives Matter and all the racism issues that are come up all at once. It's like we have three crises simultaneously.
Yes, we do. Yes, we do, and we just have to take it one day at a time.
Yes,
and then try
one day at a time. Absolutely.
Want to survive it as best as possible. Speaking of survival, what are the activities that you're doing to keep sane between writing all of these papers? How is your team doing?
Yeah, I mean, my team is, I think people are all stressing out a bit. And what I'm doing personally, I'm walking a lot. I'm kind of doing more walking than usual. My wife is really involved, and mine to a lesser extent, in playing a game called Pickleball, which is a variation on tennis, which we've converted part of our property to having 2 pickleball courts, and we have people actually come over, and we distance all the time. But we initially stopped people coming here from playing doubles, and now people only get to play singles, basically, and we have to only keep a few people here. We're really careful about it because most people are older like we are. And it's a really fun game in terms of it's a cross between tennis and ping pong and badminton. paddleball and all that stuff. And so it's played with a net outdoors. And we used to play it all here at the local Y, but the Y is shut down. So we've become, someone called us a country club where people come by now to play at our house. And we have people here almost every day. My wife has a sign-up sheet. We're developing something online. It's like,
wow.
And they were so grateful they actually bought us a brand new net to set up a second one so we can have 2 games going at once. And so, yeah, I do that not every day, but several days a week. I did it this morning. And there's some really good players, men and women, and it's fun. And it's been very useful both walking and playing that game to get rid of a lot of the tension, stress that certainly I've been feeling.
Definitely. I think we're all feeling it. And did your, just a question out there, did your dog, has your dog been surprised that you're at home more?
I don't know.
I think our dog
is so into herself. I think she's really, and my particular dog is attached to my wife. So she spends a little time with me. I try to, the last few weeks I've been trying to take her out for a walk after dinner every night. And most of the time my wife goes, but sometimes she doesn't. And she comes with me reluctantly. But I try to do that most evenings. And that's part of I'm getting on a lot of steps every day. So my average is up from last year quite a bit. So, I'm doing that. And I'm staying outdoors. I'm wearing masks all the time, of course. And as I may have told you, I have this health issue that's required me to go to get these treatments 3 times a week now. And so I'm still doing that. And in fact, I have an appointment in a half hour. I have to take this medication before I go. But so that's makes me feel really vulnerable because I've already gotten a couple of opportunistic infections related to my immunosuppression. So I'm kind of immunosuppressed. And that together with my age, together with the fact that I have asthma, I'm kind of a pretty vulnerable person. So I've been super careful about what I basically, except for going out to walk, to play that game, pickleball, and to go to the doctor, that's basically all I do. I went to pick up to the pharmacy because they wouldn't deliver the other day, and I was out of medications. But that was it. That's the only store I've been in weeks, So I'm really pretty careful.
That's very important. And then you get the time to write all these fantastic papers.
Well, yeah. But I've also been because I also I'm a co-director of an MD PhD program, and there's been a lot of energy around that from the point of view of students having stress and problems with their thesis or with their research or with getting labs or whatever, but also recently with respect to the unfortunate murders of people and the whole Black Lives Matter movement. And we put a lot of effort, and I've now gotten involved nationally with other MD PhD directors around the country who are initiating an effort around this. I've been on a couple Zooms this last few days on that. So I'm really busy, actually. I'm working harder than I ever worked the last couple of years. So it's strange in a different way, really a different way, though.
But at least you're keeping active, you're working hard, and it keeps your mind off of thinking the negative thoughts, and you're doing something great at the same time.
I hope so. I mean, the one sad thing is that I haven't gotten to see my children and grandchildren very much. We finally drove from San Diego to Los Angeles 10 days ago. For the first time, we'd seen them since March, and we distanced them and so forth. And that was really great. But that's the one thing I'm really been missing. We FaceTime, we've Zoomed or whatever, but it's not the same as person to person.
It's not. Humans are social animals, and it's not the same thing. Thank God we have the technology to keep in touch, but it's definitely not.
Right, absolutely. So.
All right. Thank you, Paul, so much for taking the time.
I hope it's useful to you. Good luck.
It was very useful. I hope people will enjoy it as well.
Okay. Hope to be in touch with you soon. All right. Bye-bye.
Bye.
Thank you for listening to this very special podcast episode. We hope you enjoyed it. We look forward to having you attend the Dr. GPCR Summit held in September. If you'd like to present your work, don't forget to visit drgpcr.com/summit-2020-explained. Thank you to Attila Forrest. Music and sound by Rosa Bershish. We also want to take a moment to thank our very talented and dedicated science communicators, Shivani Sajdev and Jean Paik. I'm your host, Dr. Yamina Bershish. Thank you for the privilege of your time, and until next time, stay safe.